Hi and welcome to this pre-recorded lesson on anaerobic respiration that I'm setting for both Year 13 groups on Wednesday. Now, I did this video already in one take, 20 minutes, but I forgot to press record. So this is the second time recording it for you. So apologies if it's not quite as enthusiastic as the first time. So first thing to do is to do this exam question over here on the right. Pause the video, give it a go, and then we'll see how you do. All right, so the first bit was about numbers of carbons. Here are the numbers of carbons: 3 carbons in pyruvate, 2 in acetyl-CoA, 6, this would be in citric acid or citrate, 5 and 4 you don't need to know the names of there. X and Y, well, Y is CO2 and X is reduced FAD. CD, the questions below. Where does glucose to pyruvate take place? That's glycolysis, so it's the cytosol or the cytoplasm. Where does the Krebs cycle take place? That's the matrix of the mitochondria. And explain how the acetyl group is carried into the Krebs cycle. I wrote it here. It's a bit difficult to run out of space there. I'll just read it to you. In the link reaction, the enzyme pyruvate dehydrogenase attaches an acetyl group onto coenzyme A. This then carries the 2C group to the first enzyme in the Krebs cycle, which joins it to oxaloacetate. That'd be 3 marks. Okay, so here's the next thing for you to think about. What is the difference between a cofactor and a coenzyme? Have a ponder of that for a second and see what you think. Okay, they're very similar words, but they mean subtly different things. So some enzymes need a non-protein substance called a cofactor to work with. Now there are 2 types of cofactor. So they can be split into activators, which are permanently bound prosthetic groups, and coenzymes, which are only temporarily bound. Okay, so that's the key thing. Both activators and coenzymes are cofactors, but permanently bound cofactors are called prosthetic groups, and temporarily bound cofactors are coenzymes that just bring in a chemical group to be used in a reaction, and then they float away. So coenzymes include vitamin C, ATP, and of course NAD and FAD. So here's 2 multiple choice questions that often come up in exams. I've seen this first question several times in a few different papers actually. So pause the video, do these multiple choice questions, and see how you do. All right, have you done that? Number 1. Cl- is the cofactor for amylase. You need to know that. And number 2, you actually do need to know this as well. This is the other one you need to know, is that zinc is used for carbonic anhydrase as a cofactor for carbonic anhydrase. And so this answer is C. So it's an inorganic ion and it's a prosthetic group, which is a permanently bound cofactor / activator. Okay, so we're going to go into a bit more detail now on some coenzymes. Okay, that was just cofactors, inorganic ions that are permanently bound. Sorry, activators, inorganic ions that are permanently bound, acting as prosthetic groups. And now we're going to look at coenzymes which are temporarily bound. So the 3 major ones that you need to know about in respiration are NAD, coenzyme A, and FAD. Now also ATP is sometimes called a coenzyme as well, so also you're going to need to write about that. Because in a minute I'm going to give you a 6-mark question on these coenzymes, which I want you to do. So NAD, CoA, FAD, and also ATP are coenzymes involved in respiration. Now briefly going to go into a bit more detail on NAD and FAD. NAD can be reduced, it can accept 2 hydrogens to form NADH plus H+. Now we can just call this reduced NAD, okay, reduced NAD. So NAD looks like this. Okay, you should be able to recognize the structure. You don't need to remember the structure and draw it, but you should maybe be able to recognize this. So NAD, first of all, it contains an adenine nucleotide from RNA because we've got ribose is the sugar. And then we've got this extra thing called nicotinamide, which actually is vitamin B3. So the whole thing is called nicotinamide adenine dinucleotide, or NAD. And that's what it is. So we can oxidize it, we can reduce it. this actually should be 2, and there should be +H+ there. So there's the chemical structure, and I'm not sure about this. You look it up if you really want to, but I think the extra hydrogen forms a dative covalent bond there, I think, is where it is. I'm thinking. So, FAD is a bit more simple. FAD accepts 2 hydrogens, so this is oxidized FAD or FAD, and then the 2 extra hydrogens go here. There's one, and this is reduced FAD, or sometimes written as FADH2. Okay, so quickly see if you can match up the molecules on the left with their role in respiration on the right. You can write that down, or you could just do that kind of verbally. Pause the video and have a go. All right, so ATP is a short-term energy store, the product of respiration. Coenzyme A aids the transition from glycolysis to the Krebs cycle. NAD+ is a hydrogen acceptor, and PI is moved between molecules to vary their energy levels. Okay? So, here's the 6th marker. I'm going to read it to you. So this question, taken from a paper a couple years ago, and the first part of the question was about photosynthesis, but then the question turns and asks you about respiration. So this question starts, the light-independent stage requires coenzymes. For example, NADPH reduces molecules by adding electrons, and ATP phosphorylates molecules by adding phosphate groups. Coenzymes are also involved in other cellular processes, including respiration. Summarize the importance of coenzymes in respiration. You should include detail of the molecules and the processes involved. So, first of all, before we get started, what are the 4 coenzymes involved in respiration? See if you can list them for me quickly. Okay, did you get them? So first of all, we've got NAD, obviously. We've also got FAD, don't forget that one. We've got coenzyme A. And finally, also, don't forget, we've got ATP. So those are the 4 coenzymes involved in respiration. So you're going to need to write detail about the role of each one and the processes that are involved, okay? The details of the molecules and the processes. So pause this video, give yourself 7 minutes to do this, or you could give yourself a bit longer. Maybe you set a timer for 7 minutes or up to 9 or 10 minutes if you want. Write your full answer and then come back. Off you go. Okay, I did that as well. So here is my answer. Now, I'm not going to read that out to you. You can pause the video and have a look at it. And here is where I've tried to highlight keywords I think are worthy of marks. So maybe pause that there, have a look at that, read it all the way through, and then compare it to your answer. So there's a lot of detail in there. That did take me around about sort of 7 or 8 minutes to write, I'd say. And let's look at the mark scheme now. So here's the mark scheme as provided by the exam board. So first of all, I want to look at the guidance, okay, and I'll come back to this slide. So the guidance says, to get 5 or 6 marks, you need that's up here, 5 or 6 marks, you need a full and detailed summary of the role of the different coenzymes, including the link reaction. Okay, so you have to have talked about the link reaction to get 5 or 6. If you forgot, you can only get 3 or 4, I think. So for 3 or 4, you need a clear summary, some discussion of their involvement with various processes, but you haven't really talked about it all. And level 1 or 2 is a limited summary. So let's look at the stuff that you can say, the actual points that you can say. Okay, I'm going to go back a slide. So first of all, for coenzyme A, here's the stuff you could say for that. For ADP and ATP, you could say all this stuff. And for NAD, you could say this stuff, and there's more on the next slide. Okay, if you need to pause there and mark your answer using that criteria, then go. Moving on to the next slide, there's more stuff we can continue to say about NAD here. All this stuff is also about NAD, and then there's this extra stuff on FAD, okay? So how many kind of content points did you say? Remember, that doesn't exactly equate to your mark. If you didn't talk about link reaction, I'm afraid you can't get 5 or 6. So hopefully you did mention the link reaction and you gave sort of 5 or 6 detail points, and that would give you 5 or 6 marks, okay? So mark your answer. How did you do? Give yourself an EBI as well. Okay, let's move on. So now we're going to get into the new content. So the new content here is all about anaerobic respiration we're going to do today. Anaerobic respiration. But first of all, let's talk about why anaerobic respiration is necessary. So respiration, hopefully in your book, you've got something a bit like this. These are all the notes that you've made on respiration as an overview. But actually, we're going to strip that away and just focus on the outline again. So what happens if there is no oxygen? If oxygen is removed, then that means that hydrogen ions can't be accepted by oxygen and turn into water, and also electrons as well. So that means that electrons build up in the electron transport chain and the whole thing stops moving, okay? Because all the cytochrome proteins in the electron transport chain will kind of become reduced and kind of clogged up. That means that these hydrogen ions carried by hydrogen carriers such as NAD, they can't be offloaded here. So we get a build-up of reduced NAD. It builds up and it builds up. Because reduced NAD builds up and there is therefore no oxidized NAD, the Krebs cycle, where you need oxidized NAD to come in and remove hydrogens, it stops as well. And in fact, so does the link reaction, because again, that needs oxidized NAD to come and take some hydrogen, so that stops as well. Now, that's a massive problem because oxidized NAD is also needed over here in glycolysis. Oxidized NAD comes in the final stage of glycolysis when triose phosphate is oxidized and produces reduced NAD. So without a supply of oxidized NAD, glycolysis would stop. So you would not get any ATP from any of these processes, and you also wouldn't get the 2 ATP from this process. So without oxidized NAD, the cell would die. So there is a solution. It's not a great solution, but it's kind of a solution. And the solution is something called fermentation. So fermentation, you could write this down, is an extra step designed to regenerate oxidized NAD so that glycolysis can occur. Okay? All right, so there's 2 ways that we can do that fermentation, depending on what type of cell, what type of eukaryotic cell you are. So eukaryotic cells either use the ethanol fermentation pathway if they're fungi or plants, so fungi and plants use that way, and mammals use the lactate fermentation pathway. So if fungi or plants, and if mammals. And I think also, it doesn't say in the book here, but I think all animals actually use that, because I definitely know that reptiles and I think birds also use lactic acid. So let's just look at those two steps briefly, and then I'm going to set you your sort of more independent work to do. Ethanol fermentation. Here we have pyruvate. So both of them start with pyruvate. Pyruvate here, pyruvate here. Now pyruvate is a 3-carbon molecule. And in the ethanol fermentation pathway, first we remove a carbon dioxide to give us a 2-carbon molecule that is called ethanal. And then that ethanal accepts hydrogen ions from reduced NAD. Here they are. They're accepting the atoms to become ethanol. So therefore, reduced NAD is oxidized to oxidized NAD. So great, we've regenerated oxidized NAD, problem solved. Lactate fermentation, similar thing. We have pyruvate, 3-carbon, but in this case, the 3-carbon molecule just directly takes the hydrogens. We don't have the carbon removed, giving CO2. So then we have lactate, which is the reduced molecule, and we have oxidized NAD regenerated again. Problem solved. Now, one extra thing that you need to know is that lactate can be used later. So if oxygen kind of returns, And in a mammal, it will, because if oxygen's not there, that's fatal. But if you just don't have enough oxygen temporarily, like if a muscle is working super hard so that it doesn't have enough oxygen to do all the respiration that it's kind of doing, it will shift into anaerobic respiration. Lactic acid or lactate will build up, causes a burning sensation in the muscles, but then it can be put into Krebs cycle. So when oxygen returns, the Krebs cycle starts running again, and lactate can be fed into the Krebs cycle to be fully oxidized to generate more energy, or it can also be converted and stored as glycogen, or in fact can be converted back into glucose. But those things require oxygen. So this generates something called an oxygen debt. If you build up lactic acid in the blood, then even if you stop exercising and you've got that lactic acid in the blood and in the muscles, your body needs more oxygen to fully break it down in the Krebs cycle, or to convert it to glycogen, for example. Okay, so your independent task now is basically just to read over this textbook page and answer all the questions. And we'll be going through the answers to those questions next lesson. So make sure you've done all of this by tomorrow's lesson, which is Thursday. All right, that's the end of the lesson, and I'll see you on Thursday. Bye-bye.